Today we are going to discuss about the subject virology and parasitology. In that, myself am Mrs. Manonmani, Assistant Professor, Department of Microbiology, SFR College, Sivakasi. In virology and parasitology, today we are going to discuss about Paragonimus westermani. Now we are discuss about some small introductions about Paragonimus westermani. These parasites was first discovered by Kerbert in the year of 1878 from the lungs of Bengal tigers. These animals which was died in zoological gardens. From that we can collect the sample for analysis. This is one of the endemic parasitical diseases. These type of parasites mostly found in Far East, including Japan, Korea, Thailand, China, Southeast Asia, Sri Lanka and our India. The Paragonimus westermani, namely commonly called as Oriental lung fluke. So, this is the morphological appearance of Paragonimus westermani. In adult stage it is egg-shaped organism, about to have 10 micron millimeter length, broad and 4 millimeter in thick size, and which is reddish, red brownish in color. In their anterior end, one of the oral suckers was placed, and their posterior end and ventral suckers was placed, which was located towards the middle of the body. So, the overall life cycle or lifespan of Paragonimus westermani in human body is up to 20 years. So, what are the life cycles of Paragonimus westermani? So, human during their life cycle, the parasites can act as defined host, primary host and secondary host. So, in which human acts as a defined host. So, humans acquired infections by eating the undercooked crab or gravy fish containing Metazirgazeae. So, Metazirgazeae, one of the stages of Paragonimus westermani. The adult worms can live in the respiratory tract in the defined host of man, like man or cats or tigers, pigs, dogs, etc., foxes, etc. So, they are the defined host for the development of adult worms Paragonimus westermani. Unembryonated egg can escape into the bronchi, and which was again passed out through the feces or swallowed with the sputum. Get matured, it will take times for 2 weeks, and then these type of matured eggs can release us into the water and move into the free swimming in the form of myriazidia. So, these myriazidia can infect the first intermediate host like snail, freshwater snails. So, from the defined host, the organisms or this parasite can involve with the help of free swimming in the water, then it reaches the intermediate host snail. So, in that snail the Sergaceae was formed. It is also one of the stages of parasites, and these which was released from the snails after several weeks along with the development of short stumpy tails. So, then Sergaceae swim in the streams and enter the gill chambers of the second intermediate host like grey fish or crabs or gills of the crabs, etc. So, then further stages was development and encysmation process will take up within the gills of the crabs or grey fishes only. When the defined host that like a human or frogs or foxes, when eaten these infected grey fish or crustaceans, through this which is travel into the in our body and develop the symptoms, and the life cycle again and go through it. So within the human host or men host, the metazoraceae were developed in the part of duodenum within a few minutes after inversion. Then they migrate through the diaphragm and reaches the lungs, and in that place the organisms will settled within the bronchiole and develop into adult worms will take 2 to 3 months. In that the parasites can develop for the infectious stages and causes infections to the human beings or defined host. So in rare cases, some type of Paragonimus vestimonis can reach the brain and also causes severe diseases. So during their fertilization or maturation, these type of parasites need 2 worms for their fertilization, even though it is a hermaphroditic. So these are all the life cycle of Paragonimus vestimoni. Half of the life cycle which was carried out in the water with the help of their free swimming capacity. So first intermediate host is snail, second intermediate host is crab or fishes, and we are the final host, namely called as defined host or infectious host. So what are the clinical features of Paragonimus vestimoni? Abdominal pain is a common, and also diarrhea, fever, abscess formation, pneumonitis, Paragonimus. In its brain, special name is called as cerebral Paragonimus. So what are the laboratory diagnosis of Paragonimus vestimoni is microscopic examination. Based on their morphological appearances, we can easily find these parasites with the help of microscopy. Then serological test with the help of blood collections or serum samples, we can easily find, or antigen antibody reactions, we can easily find the pathogens from the particular hosts. Example, complement fixation test, ELISA test. So imagine captures with the help of chest X-ray or CT scan of the chest. Why we are choosing the chest is microorganisms that these Paragonimus vestimoni can develop within the defined host within the bronchi, so that we can easily find the microorganisms in the chest part. So what are the prophylaxis of Paragonimus vestimoni? Prasic and 12. So it is the first and one most drug of choice for the treatment of Paragonimus infections, and this type of tablet per kg 25 milligrams will be suggested 3 times daily for 1 or 2 days will be taken, and other drug of choice is bitenol and nicloflan. These are all the effective drugs for the treatment choice or options for the Paragonimus vestimoni infections. Then how can we control or prevent the Paragonimus vestimoni infections to human? So first one is we should adequate cooking or proper cooking of crabs or grey fishes, which is essentially to prevent the infections. Then hands should be washed thoroughly after preparing the crabs or grey fish for food. So treatment of infected persons reduce the transmission. So if we find persons who are suffered with Paragonimus vestimoni, they are also undergo a proper treatment. These are the only choice for the transmission of these diseases. So disinfections of sputums or fecals, which may also help to control the spread of Paragonimus vestimoni parasites. So because why we are using disinfections as sputums or fecal only, in fecal samples we are having a lot of Paragonimus vestimoni parasites in it, or sputum while coughing or sneezing, from the lungs the organisms will be upheld along with the sputum samples. Then so eradication of some seasonal foods also recommended for the prevention of Paragonimus vestimoni disorders. Thank you.